Hello, this is Transport in Plants. This is Ms. Foglia's PowerPoint narrated by Dr. Meade. For my current class, we started this yesterday, but I'd like us to start from the beginning because I plan on using these videos again. Okay, we've talked about this before, about what is transported in a plant, and we've talked a little bit about how, but we're going to take it down to the cellular level. Alright, so water and minerals are coming up through the roots. The process is being driven by transpiration, but we're also using the other properties of water, cohesion and adhesion as well. Of course, water and minerals are transported in the xylem. There's that. This creates negative pressure. The negative pressure is also called tension, and the tension and negative pressure is created by the transpiration in the leaf that pulls the water up. So that's a little bit complicated to understand, but just remember that it's about negative pressure or tension for the xylem, which is different than for the phloem. So sugars are transported in the phloem, okay? The Calvin cycle loads the sucrose into the phloem so that the plants can use those sugars. The sugars can go up or down as they're needed, but primarily they go down. And this is a different kind of pressure. It's a hydrostatic pressure called positive pressure that's created by the movement of the sugars in the phloem. That's what the bulk flow causes. So we're moving sugars around. That's that process called translocation. And when we do translocation, it creates positive pressure. So the movement of the sugar is mostly down. And gas exchange is photosynthesis, of course, and cell respiration. So with photosynthesis, carbon dioxide in, oxygen out. For respiration, oxygen in, carbon dioxide out. Plants do it all. I always miss whatever the last thing is. I think I'm moving on to the next slide, and I'm not. And it's that silly penguin. So why does overwatering kill a plant? How about if you can give me a good answer for that, I will give you 2 points on your next quiz. All right, so as I was explaining in the other slide, the ascent, going up, of xylem fluid is caused by transpiration. So when we release water out of the leaf, the water starts being driven upwards. Okay, water and minerals in the soil. Water is going into the roots, okay, and into the cells with osmosis and aquaporins. Remember aquaporins from when we were learning about osmosis? Aquaporins are specialized proteins that are channels to allow water into a cell. All right, good. There's the picture. Okay. So let me get it all out here, and then it's a little easier to talk about. Osmosis is passive transport, so water can enter cells with passive transport. Minerals, however, have to enter with active transport using proton pumps. the active transport of protons. Hang on, I just want to see if the next slide goes into more detail with this. I forget. Hold on. Sorry. There we go. Oopsie. Sorry about that. So yes, the next slide goes into more detail and the pictures are a little bit bigger. So I'll give you the basic points here and then it moves on. So the root hairs on any root are there so that water will move into the plant by osmosis through the aquaporins. So here's a picture of aquaporin. It's just a little channel. It's a protein in the membrane to allow water through it. The minerals are absorbed through active transport using proton pumps that create a change in the charge so that minerals can be drawn in. And so this picture is showing you root hairs. So water, this blue dot is water, is coming in and it's going through a couple layers of the outer layers of cells in this plant and going into the xylem to go up. Okay. So mineral absorption with the proton pumps. Okay, so the active transport of hydrogen ions, protons, out of the cell, okay, which is chemiosmosis. I'm sure you remember that word. So we create a proton gradient. We create a change in the charge, which creates a membrane potential. So the inside of the cell is negatively charged because it has pumped out hydrogen ions. Let me say that again, that's a big point. The inside of the cell is negatively charged because it has pumped out hydrogen ions. So there's an imbalance of charge, and the cell wants to be neutral, so it's going to take in cations. By taking in cations, you can take in minerals. So the negative ions sneak in with the positive ions. That's co-transport. Okay, so here's the rest of that. So it's the co-transport. So protons are being pumped out. It's creating a negative charge in the cell so that the cell really wants to take in more positive charge. So here's potassium going in. And another thing that can happen is that what it says right here, the cell accumulates anions, for example, by coupling their transport to the inward diffusion of protons through a co-transporter. So they sneak in. So we create a concentration gradient and we create a charge so that cations can go in and other things, anions, sneak in with some protons. And that's co-transport. Okay, so here let's talk about how water flows through the root. Okay, one thing that you may not realize about cell walls, which is kind of cool, is that water can flow through the cell wall and not enter cells. It can hang out in the cell wall. And plants need to find a way to force the water into cells. So water has two methods of flowing through the root. Root hair is a modified cell, and so the pink and blue here are showing the two ways that water can enter. We have apoplastic and symplastic. So apoplastic is shown here in pink and symplastic is shown here in blue. And the apoplastic root, we end up having water go into the cell wall and then it has to be forced into the xylem, whereas the symplastic root, it just goes on in. Alright, so we're going to see this in some more detail. But what I want you to take in from this slide is that we have these two different roots, okay? And the whole point is to get from the soil through the root hair, through the dermis, through the cortex of the plant, and through the endodermis to get to this vascular tissue, the xylem vessels, right? Because these cells are not going to use the water. These cells are, okay? And the pink, as you can see, oh, it goes around the cell walls because it's water in the cell walls, okay? And this one, the blue, yip-de-doo, it goes through the cytoplasms of all these cells, and it kind of makes its way through little pores in the cell wall and comes right in. All right, and so the answer to this particular issue is called a Casparian strip. All right, so it is a strip between the two cells of the epidermis, and it will allow water to be forced into the cell. So here again is that same picture, and what happens is that when the water, or when the apoplastic root, that the water hits the Casparian strip and goes into the xylem. And that's the gist there. So what do you need to know? You need to know that there's two roots for water, the apoplastic root and the symplastic root. And I would remember that symplastic is the simple one. It goes on in, goes through cytoplasm, and goes in. Apoplastic is the more complicated one, and it's got to use these Casparian strips in the endodermis to hop into the xylem. All right, controlling the route of water in root. Isn't that fun to say root in two different ways in one sentence? Haven't you just been living for that moment? I'm sure you have. The endodermis, the cell layer surrounding vascular cylinder of the root, lined with the Casparian strips, and it forces fluid through the selective cell membrane. And it acts as a filter. So it can leave out what it doesn't want. Here comes a penguin. Structure and function yet again. So of course, the structure of this strip, I think is what they're talking about, does what it looks like it would do. So let's look at it, cross-section of root, monocot and dicot. And, okay, there's the area where you would have the Casparian strips. Right, so in the dicot, it's right here. In the monocot, it's right here, okay? Because you have the vessels right here, right? So the Casparian strip's going to be near the xylem. Okay, another way that root hairs can absorb more is by having this symbiotic relationship with a fungus, Mycorrhizae. So it increases absorption. So these fungi hang out on the roots, and they're happy to hang out there because they benefit from the structure of the plant so that they have some place to be, and they add a whole bunch of absorption. And some of their the fungus branches kind of push in and are in some of the cells, and it helps the absorption of nutrients. So it's symbiotic fungi, greatly increasing the surface area for absorption of water and minerals. Oh, oopsie. Increases the volume of soil reached by the plant, and increases transport to host plant. Right, so if it's a way that the plant gets to extend itself out a little further. All right, so here you can see, here's the epidermis, the cortex, and the endodermis of the plant, And you can see the fungus, kind of the purple, that it's worked its way in pretty far, right? So it might be pretty far out of the plant and then works its way in a little bit. And so these sort of branches, okay, are called hyphae. Are they in this picture? Okay, but just for your information, it's called hyphae, Hyphae, Hyphae. Okay, and that's where the extensions are. It's been seen that with some more recent studies that uh, that the fungus helps with phosphorus absorption most of all. Phosphorus is hard to get in a plant, and this helps really improve the amount of phosphorus we take in. So that's the main point of that. The transport of sugars in the phloem. Okay, we were just talking about the minerals and the xylem. Okay, the loading of sucrose into the phloem, it flows through cells via plasmodesmata. Do you remember what plasmodesmata is? I'm hoping you do. And if you don't, look here, handy picture. So it shows you it's kind of the little passageway in between two cells. So it's a way two cells can communicate with each other. All right, and we have proton pumps again, creating gradients. So we can have co-transport of sucrose into cells down the proton gradient. So these proton pumps are really important for creating a significant change in the charge of the cell and affect the action potential of the membrane so that stuff can come in. And so how do sugars get into the phloem and be transported? Well, they use this proton gradient to create, or the proton pump to create a gradient, and then we can co-transport some protons along with some sucrose. Okay, so this little protein is a proton pump, and this protein is a co-transporter. So sucrose gets to ride along with some protons to get inside, and that again is co-transport. All right, so we said it was positive pressure in the phloem. Okay, there we go. So phloem can flow at a rate of 1 millimeter per hour. And remember that xylem can only go from roots to leaves, but phloem can go wherever it's needed, up or down. So sieve tubes carry sugars from a sugar source to a sugar sink. A sugar source is a plant organ that produces sugar by photosynthesis or breaking down starch. Mature leaves are the primary sugar source. A sugar sink is an organ that's a net consumer or storer of sugar. Growing roots, buds, stems, and fruits are sugar sinks. So in this case, our sink cell is right here, which is going to store the sucrose. All right, so this blobby cell could be a cell for a root or a bud or a stem or a fruit. All right, somewhere to store sugar. So you think of fruits as deliciousness. Well, they're also a handy way to store a lot of sugar. A bulb, okay, or a tuber can be a source of having a sink. The sugar sink, like whatever kind it is going to receive sugar from wherever the nearest source is. So it's not going to needlessly travel far. If there was a leaf right here doing photosynthesis, then that would be, oops, that would be the leaf supplying sugar to this sink. Okay? Um, it is an active transport process to get sugar into the phloem. Okay? I don't know why we have this bit about xylem here, but anyway. Okay, so the key thing that the penguin is pointing out to you is to know what a source and what a sink is. So a source is something like a leaf that does a lot of photosynthesis and produces sugar, or a cell that does a lot of breakdown of starch. And a sink is a fruit or a bud which can store sugar. All right, experimentation. Oh yeah, we get to skip this right now. All right, we're not going to talk about this right now, but if you love maple sugar, which I totally do, this is how they get it out. The phloem of maple tree. All right, so that is it for transport. Don't you love it? 21 minutes. Hang on, I'm going to give you a little question sheet on transport so that you know what the main points are to review. Okay, before I get to the questions, there's a slide here that I wanted to show you that I forgot about, is just to talk about the control of the stomata, or as she says, stomates here. So this is the anatomy of it. So these are called guard cells, surrounded by epidermal cells. So the guard cells are the key here. They're what opens and closes. All right, so the uptake of potassium ions by the guard cell happens by proton pumps. Water enters by osmosis and makes the guard cells become more rigid, turgid, full, firm. The loss of potassium ions by the guard cells, okay, the water leaves by osmosis and the guard cells become flaccid. So they go back and forth between being opened and closed, and that's how they do it, is by the transport of potassium ions and water back and forth across the guard cell from the epidermal cell. Okay, so water moves into the guard cell, water moves out. So when water moves in, it gets turgid and it's open. When it moves out, it's flaccid and closed. All right, so this has some real pictures of them. So you're always trying to find, as a plant, this balance between photosynthesis and transpiration. So a leaf may transpire more than its weight in water in a day, and this loss must be balanced by the need for carbon dioxide for photosynthesis. Okay, now we'll do the questions. All right, here's a couple questions. What I want you to do is for somebody to, after I read the question, I'll say pause here. You pause. When you unpause, I'll tell you the answer. Most of these questions come from that site that I keep giving you from Biology Junction, where there's a list of, hundreds of sample questions for each chapter, or each unit rather. Okay, hyphae form a covering over roots. Altogether, these hyphae create a large surface area that helps to do which of the following? Aid in absorbing minerals and ions, B, maintain cell shape, C, increase cellular respiration, or D, anchor a plant. Oops, I forgot to move E here. and E, protect the roots from ultraviolet light. So pause now. Okay, the answer is A. Next question. Phloem transport of sucrose can be described as going from source to sink. Which of the following would not normally function as a sink? Pause now. Okay, the answer is a mature leaf. And the reason for that is a storage organ in the summer would be like a fruit. And these are all actively growing places, so that's the perfect place to be using sugars. Okay, a mature leaf no longer needs a whole bunch of sugars to grow. Which of the following is a correct statement about sugar movement in phloem? Diffusion can account for the observed rates of transport. Movement can occur both upward and downward in a plant. Sugar is translocated from sinks to sources. Only phloem cells with nuclei can perform sugar movement. Sugar transport does not require energy. Pause now. Okay, the answer is B. So that's the only one that's true here. Right? Diffusion has to do with xylem. Sugar is translocated from sources to sinks. This is not true because the phloem cells that do the sugar movement do not have a nuclei, and sugar transport indeed requires energy. Okay. What is the main force by which most of the water with xylem vessels moves toward the top of a tree? Active transport of ions into the steel, atmospheric pressure on roots, evaporation of water through stoma, the force of root pressure, or osmosis in the root. Pause here. All right, the answer, of course, is C. The transpiration, the evaporation of water through the stoma, is the main thing that is driving the tension, the negative pressure of water up a tree in the xylem. Sucrose enters a phloem sieve tube cell because of osmosis, water potential, active transport, or a process regulated by auxin. Pause here. Okay, and the answer is C, active transport. All right, this has come to an end. I hope it's been helpful, and feel free to add more to our plant review now that you've reviewed the plant transport. Bye, guys.